Let's climb into our Wayback Machine and go way back to the beginning of time, or at least to the time of our conception. Here you're viewing the female reproductive tract. This is an ovary. Inside the ovary are follicles containing eggs. This is a uterine tube, or fallopian tube. And this is the uterus, or womb. Note that a follicle has ruptured and released an egg, which has been sucked into the uterine tube. The egg is greeted there by a gang of sperm. Oh, happy day! One of whom penetrates the egg, delivering its 23 chromosomes to the 23 that are in the egg. The result is a fertilized egg, or zygote. From that point on, three major processes will occur that will result in the development of a multicellular organism. Those processes are cell growth, cell division, and cell differentiation. Let's examine each. Cell growth usually occurs before cell division. But why? Suppose you bought a small pie, but to stay slim, you invited a friend to share it with you. Unbeknownst to you, your friend invited a couple of her friends, and these people invited a couple of their friends. And now there are 8 hungry people standing around your modest pie waiting for a piece. You decide to forego a piece because it would only make you more hungry. A similar principle applies to the cell. When a cell divides, the offspring are half the size of the parent. If they divide without growing, their offspring will be a fourth the size of the original. If these cells divide without growing, their offspring will be an eighth the size of the original. You can see that cell division without growth can result in cells dividing themselves into nothingness. And that's no place to be. Cells need enough volume to support their organelles. Eukaryotic cells can be too small to perform their necessary functions or to survive. Cell division refers to the process of a cell splitting in two. First, the cell prepares to divide by replicating its genetic information. Then it splits into two, with copies of each chromosome going to each offspring cell. These new cells may themselves go through the process of division following growth, so we have a repetitive process called a cycle. But why do cells divide? The fertilized egg could more simply grow until it was adult size. Most organisms on Earth are unicellular, so why aren't we? Part of the problem has to do with the needs of cells to get nutrients into the cell and waste products out of the cell through the plasma membrane. When a cell grows in volume, so does its surface area. However, the volume of the cell increases at a faster rate than does its surface area. So the ratio of surface area to volume declines. This decline in surface to volume ratio limits the ability of a cell to meet its needs for nutrients and removal of waste products. The solution for creating a larger organism is for cells to divide once they've reached a critical volume. When a cell divides, it restores a more favorable surface area to volume ratio. If you cut your apple in two, do you still have the same volume of apple to eat? Sure you do. What have you changed? You increased the total surface area. If you turned your apple into applesauce, it would have an enormous surface area to volume ratio. That is why unicellular organisms are microscopic. It is simply not practical for a single cell to be our size. The cell cycle refers to the series of events a cell uses to prepare to divide, and then to complete the division. Cells spend most of their lives in interphase. Interphase is when cells do their chores. They grow, they actively transport substances across membranes, they make ATP, they recycle cellular parts. If a cell is going to divide, it must prepare to divide by replicating its genetic information. Once this is complete, the cell is ready to enter the division phase of the cell cycle called mitosis. Here's a graphic of the cell cycle. Interphase dominates the cell cycle. In fact, many cells, like nerve cells and muscle cells, are in permanent interphase. They no longer will divide. We call them post-mitotic. Those that can divide replicate their genetic information during interphase and then enter mitosis, the phase where one cell splits into two. Here you're viewing a map of an interphase cell before it has begun to prepare to divide. The chromosomes are single-stranded. Here is the cell after replication of the chromosomes. The chromosomes are now double-stranded. Each strand is called a chromatid. When the cell divides, the chromatid pairs will separate and end up in different cells. Chromosome replication requires replication of the DNA within each chromosome. To replicate the DNA, the parent strands are separated, and the enzyme DNA polymerase walks along each strand, reading the sequence of nucleotides and constructing new complementary strands. The end products of each chromosome are two double-stranded DNA molecules that should contain the same information. Here we have a representation of a double-stranded DNA molecule. Let's watch DNA polymerase replicate the two parent strands. Recall that in DNA, thymine always pairs with adenine, and cytosine always pairs with guanine. T with A, G with C. When the enzyme is finished, we have two double helix molecules that should contain the same sequence of nucleotides. All information is conserved. Each double-stranded DNA molecule will be contained within its chromatid of a double-stranded chromosome. When the cell divides and distributes the genetic information, both offspring should get the same information. They should be genetic clones of the parent cell and each other. Here is a replicated double-stranded chromosome. It was replicated during interphase. Each strand is called a chromatid. The two chromatids are held together at their centromeres. Within each chromatid is a double-stranded DNA molecule. Look closely, and you can see that each DNA molecule has the same sequence of nucleotides. So when the chromatids separate during mitosis, each cell will get all the genetic information present in the parent cell. Mitosis is when a cell and its double-stranded chromosomes literally split in two. Mitosis is relatively brief, taking only about a half hour to complete once it begins. Mitosis is usually subdivided into shorter phases, each characterized by distinguishing events when viewed through a microscope. Mitosis is usually subdivided into four phases: prophase, metaphase, anaphase, and telophase. it might be helpful to remember the acronym PMAT. Let's see what is unique about each of these phases. Here is the view through the microscope of a cell transitioning from interphase to the first stage of mitosis, prophase. Thanks to staining, we can see the plasma membrane, the nuclear envelope and the nucleus, and the cytoplasm of the interphase cell. What do you notice about the changes in prophase? Note how the nucleus has darkened. What has happened is that the chromosomes are condensing in preparation for transport to their new home. To get information out of your textbook, you have to open it. But when you're transporting your textbook to another location, you close it because it's easier to carry. Likewise, when the cell is in interphase, it needs to read the information in the chromosomes, so they must be unwound. But when the chromosomes need to be transported, they are condensed. Our genetics books are closed, and so our chromosomes are more easily seen in the cell. When the chromosomes are condensed, however, the cell can't access the information within, which is one reason mitosis must proceed rapidly. Note also there are two centrosomes in this prophase cell. The centrosome was replicated during interphase along with the chromosomes, and the two centrosomes are now at opposite poles in the cell, preparing to separate the double-stranded chromosomes. Recall that the centrosomes are central for cell movement, including movement of the chromosomes during mitosis. By the end of prophase, the nuclear envelope is gone, disassembled to make transport of the chromosomes easier. So technically, at the end of prophase, there is no more nucleus. Here you're viewing the transition between prophase and metaphase of mitosis. Can you describe the obvious differences? In metaphase, the chromosomes have formed a single line in the center of the cell. This line of double-stranded chromosomes was created by the two centrosomes, which sent out microtubules, known as spindle fibers, to attach to the centromeres of each chromosome in order to align them. Next, we see the transition from metaphase to anaphase. What appears to be new in anaphase? It appears the chromatids of the double-stranded chromosomes have been separated by the two centrosomes. In a human cell, this would be two bundles of 46 single-stranded chromosomes migrating to opposite poles of the cell. Here is a drawing of the two centrosomes with their microtubules attached to the centromeres of the chromatids after separating them. When you see two distinct bundles of chromosomes in a cell that appear to be moving in opposite directions, you're viewing anaphase. Our last phase of mitosis is telophase. Here you're viewing the transition from anaphase to telophase. What do you notice is different in telophase? First, it appears that the two bundles of chromosomes are no longer being pulled apart. They've reached their destination. Also note the pinching off of the cytoplasm, known as cytokinesis, and new plasma membrane forming between the offspring cells. When the two bundles of chromosomes unwind and disappear, we will be back in interphase, except now we have two cells instead of one. Telophase is usually the toughest phase to find on a slide because it's the shortest phase. Here is a cell in interphase. Here's the plasma membrane. The blue is the cytoplasm. The yellow is the nucleus. And this organelle is the nucleolus. Note that the chromosomes are not visible. The chromosomes are distended so that the information within them can be read to make protein and also to replicate the genetic information. Now let's start the process of cell division. The chromosomes are starting to condense. We're entering prophase. Now the two centrosomes are lining up the chromosomes single file in the center of the cell. We're entering metaphase. Now the chromatids will be separated by the two centrosomes, pulled to opposite poles of the cell, anaphase. When the chromosomes reach their destination, we enter telophase, which involves also cytokinesis and the manufacture of new membrane between the two cells. So far, we've studied cell growth and cell division. Our next topic, on our way to becoming a multicellular organism, is cellular differentiation. If a bacterial cell fell into a nutrient agar in a petri dish, it would take advantage of the picnic environment and start replicating. Within 30 minutes, there would be 2 bacteria. In another 30 minutes, 4, then 8, then 16, 32, 64, 128, and eventually millions of bacteria that form a visible colony in our petri dish. To deal with this population explosion, we call in the Navy SEALs to have them blast away at the bacterial colony, destroying 90% of the cells in the colony. What happens to the remaining cells? Do they weep? Do they die? No, most likely they party. The Navy SEALs did them a favor by wiping out their siblings. Now they don't have to share nutrients with them. What would happen to your surviving cells if the Navy SEALs blasted to smithereens 90% of your cells? Your surviving cells would soon join their deceased brethren. Why is the outcome different for us than for a colony of bacteria? The difference is that we are a multicellular organism, whereas a colony of bacteria is a colony of unicellular organisms. Bacterial cells don't depend on each other for survival. Our cells do. Even the loss of a few cells in our body can result in the death of all. The beta cells of the pancreas represent less than 1% of all the cells in our body. Yet if they died off, the rest of our cells would soon follow without medical intervention. The survival of our brain depends on our heart. The survival of our heart depends on the survival of our liver. The survival of our liver depends on the survival of our kidneys. It's a similar situation in our society. Each of us depends on farmers, doctors, plumbers, auto mechanics, trash collectors, carpenters, etc. to stay alive and prosper. If the trash collectors went on strike, we would soon be knee-deep in filth. We depend on each other for our survival just as our organs depend on each other for their survival. During the first couple of weeks following conception, our cells are undifferentiated. They all look pretty much alike, except they may be in different stages of the cell cycle. Then cells begin to differentiate, to specialize. Some become blood cells, some become muscle cells, others become nerve cells. You're viewing some of the different kinds of cells in our bodies, all of whom descended from the same fertilized egg. On the left, you have a skeletal muscle cell. These cells tend to be long and slender. In fact, oftentimes they're called fibers instead of cells. Skeletal muscle cells are striped, and they have multiple nuclei. And their primary function is to contract, to generate a force called tension. Next, we have a smooth muscle cell, so named because it does not have the stripes that are present in the skeletal muscle cell. Smooth muscle cells have a single nucleus. They're oftentimes called involuntary muscle because we normally do not have conscious control over their contraction. The third cell is a nerve cell, or neuron. Nerve cells can be very long. I have nerve cells that begin in my foot and go all the way to my spinal cord. Their primary function is to conduct information. They are the wiring system of our body, allowing us to rapidly communicate from one part of our body to another. The fourth cell is a sperm cell. This is a gamete, a sex cell. The sperm cell is unique in that it has a flagellum or tail that gives it mobility so it can find an egg. This is the only cell that humans produce that have a flagellum. If you're a female, you're just out of luck. The head of the sperm contains 23 chromosomes. When it finds an egg and fertilizes an egg, the normal 46 chromosomes will be restored. The fifth cell is called a goblet cell. We find this cell in mucous membranes of the body. The cell produces mucus, that's snot to you. As mucus accumulates inside the cell swells and resembles a goblet, and then it exocytoses the mucus to the free surface. The red cells are called erythrocytes, which is Greek for red blood cell. The red blood cells circulate in the blood. Their primary function is to transport oxygen from the lungs to the other organs of the body. Red blood cells are rather unique in the sense that they do not have a nucleus. They don't even have mitochondria. But they are full of a protein called hemoglobin, which is necessary for the function of the red blood cell. This next cell is called an astrocyte, a star cell. It's a support cell within the central nervous system, an example of what are called glial cells. And finally, the two cells you're viewing here at the bottom are examples of epithelial cells. These cells can be found on surfaces in the body. This particular epithelial cell has hair-like extensions called cilia. We'll find these, for example, along the respiratory tract. Debris that you inhale that gets trapped in the mucus of your respiratory tract can be swept toward your throat so you can either swallow it or spit it out. So these are just a few of the specialized cells that can be found within our body, and all of them originated from the same fertilized egg. The process of generating these specialized cells from an undifferentiated egg is what we call cellular differentiation. Let's look at the mechanism of cellular differentiation. Do cells change their genetic composition when they differentiate? Do they lose genetic information? It appears the primary means of differentiation is selective activation of genes. In other words, nerve cells have the same 20,000 genes as muscle cells and endocrine cells. But in nerve cells, the muscle genes are switched off. And in muscle cells, the nerve cell genes are switched off. Cells of the adrenal medulla contain the gene for insulin. But in these cells, the insulin gene is switched off. And the genes for making epinephrine are switched on. It's the opposite in beta cells of the pancreas, where the genes for making epinephrine are switched off, and the gene for making insulin is switched on. The fact that specialized cells retain the genetic information present in the fertilized egg means potentially they could be reprogrammed to create entirely new organs or even entirely new individuals. In 2004, a team of researchers reported successfully reprogramming mature cells from a fruit fly back to their youthful, undifferentiated state. You are viewing a map of the skin, which is composed of two layers. The deep layer is the dermis. The outer layer is the epidermis. The dermis is where the blood circulates. The epidermis has no blood vessels. If you cut yourself superficially, you don't bleed. If you cut to the dermis, you will bleed. The outer layer of the dermis is composed of dead cells. It has to be that way because these cells have been pushed a long way from their source of moisture and nutrients. But even in death, they help seal moisture in and keep germs out. However, these dead cells are constantly falling off the surface. Those particles you see floating in the air when sunlight passes into your room are dead skin cells from you, your relatives, and pets. So show some respect. We must replace these cells continuously, and that is the job of the cells in the deep layer of the epidermis. The deep layer contains stem cells capable of cell division. Stem cells are relatively undifferentiated cells, kind of like children. When you are young, all you want to do is play. Eventually, though, your parents evict you, and you have to find an occupation. You have to differentiate. When stem cells of the epidermis divide, one of their offspring migrates to the surface, differentiates, and then dies, eventually falling off. The other sibling, however, remains in the deep layer to serve as a source of more cells in the future. Somehow we manage to produce just the right number of new cells to replace the old. And if we cut our cells, the rate of cell division increases until repairs have been made, and then the rate returns to normal. It's apparent that cell division must be regulated. Stem cells are receptive to messages from their environment that tell them to divide more or less rapidly. When these cells begin to ignore the signals to stop dividing, we may have a potentially serious disease. These were the 10 leading causes of death in the United States in 2019. Heart disease has been the leading killer in the United States every year since 1920. Cancer has been the second leading killer every year since 1938. This table says that 21 out of every 100 deaths in the U.S. in 2019 was from cancer. One of the characteristics of cancer is out-of-control cell division. This is what happens when you produce too many cells. We call excessive cell division hyperplasia, or proliferation, and the result is a cellular tumor. Most cellular tumors are benign. They include skin tags, moles, polyps, nodules, and calluses. But some are more serious, even deadly. The technical term for potentially deadly tumors is malignant. This includes cancerous tumors. Another characteristic of cancer tumors is dysplasia, or chaotic arrangement of the overgrowth of cells. Notice how organized the cells are in the column on the left. In the middle column, we have hyperplasia, excessive production of cells, but the cells otherwise are well organized. In the third column, not only do we have hyperplasia, but the cells are disorganized, which is a characteristic of cancer cells. Dysplasia. Moreover, these cells may not function normally, a condition known as anaplasia. Leukemia is a cancer of cells in the bone marrow that produce white blood cells. Leukemia means literally white blood, referencing the elevated count of white blood cells, which are responsible for defending the body against infection. You would think that with an elevated white cell count, known as leukocytosis, you would be more resistant to infection. Yet, leukemia patients are less able to fight off infections. This is because the white cells that circulate are immature and haven't the slightest idea what they're supposed to be doing. They just get in the way of other cells that could be useful in fighting off infection. The third and most deadly characteristic of cancer is metastasis. Metastasis means a tendency to spread. Localized tumors usually don't kill. That's because the neighboring cells are glued together by various types of intercellular junctions. The production of these junctions, like you see here, is promoted by vitamin D. But if these cell junctions are weak, perhaps due to a deficiency of vitamin D, the cells can break away from the primary tumor and spread throughout the body. This is what we mean by metastasis. As a result, treatment becomes more complicated and the prognosis for recovery less favorable. Here is a primary tumor. Note that cells have escaped from the primary tumor and with the help of the circulatory system are being spread throughout the body, where they've established secondary tumors. When this happens, we say a person has stage 4 cancer. Stage 5 is the funeral. Most skin cancers do not kill. In part, it's because they can be detected early and easily treated. It's also because most skin cancer cells have difficulty entering the circulatory system. However, the third most common skin cancer, melanoma, is the one most likely to cause death. That's because it's the one most likely to metastasize. People don't die of tumors on their skin. They die when these tumors shed cells that migrate to the lungs, brain, liver, or other vital organs and set up secondary tumors. Melanocytes are cells that produce the dark skin pigment, melanin. If you develop a pigmented tumor on your skin, or if a mole begins to change shape and bleed, you should get to your doctor for evaluation before it has a chance to metastasize. We know that agents that can mutate your DNA can cause cancer. Radiation, like X-rays and ultraviolet, can damage DNA. Some chemicals can damage DNA, including benzene from tobacco smoke and acetaldehyde from alcohol consumption. Viruses such as hepatitis C and papillomavirus can damage DNA and cause cancer. Usually, it takes a significant or prolonged exposure to a carcinogen to trigger cancer. Smoking a cigarette a day is not likely to affect your cancer risk, but a pack a day for many years most certainly will. The popular conception of cancer is this first curve on this graph. You get cancer, the cancer spreads rapidly, becomes symptomatic, and kills, all in a very short period of time. You might even develop stage 4 cancer between yearly screenings for cancer. These cancers are called the birds. They're hard to restrain. Other cancers kill more slowly, taking years to dispense their grisly fate. They're the rabbits. This is true of most deadly prostate cancers. Because of their slow progression, they can more easily be detected and treated. But many cancers don't kill, even if left untreated. The individual dies of something else before the cancer can kill you, or the cancer stops growing, or may even disappear. There is much evidence that cancer arises routinely in our bodies, but the vast majority of these cancers stop growing and may even disappear without harming the patient. One of the functions of our immune system is to detect and destroy abnormal cells. It is not easy to predict the natural progression of a cancer, but if you go for asymptomatic cancer screening and a small mass of cancer cells are detected, you most likely will be advised to seek treatment instead of watchful waiting. This has led to a modern phenomenon known as cancer overdiagnosis and overtreatment, and has resulted in many people being needlessly alarmed and harmed. Cancer may be overdiagnosed and overtreated if the abnormality is asymptomatic, if it will never progress to cause symptoms, and if the person will die of other causes first. Prostate cancers are among those that are frequently overdiagnosed and overtreated. This chart shows the incidence of prostate cancer on autopsy in men who died of other causes. Note that most men over 60 years of age have evidence of prostate cancer, yet only 3% of men die of prostate cancer. This suggests that most men die with prostate cancer, not because of it. But if you get a screening diagnosis of prostate cancer, the pressure will be intense to get it treated. And the side effects of treatment are significant, including erectile dysfunction and urinary incontinence. Some men die of the treatment. In the 1970s, President Ford's wife was diagnosed with breast cancer. She began a crusade to encourage asymptomatic women to get annual screening mammograms for cancer. Her campaign was very successful, as you can see here. There was an enormous growth in the diagnosis of early-stage breast cancers. With all these additional early-stage breast cancers being detected through screening, one would think we would see an equally dramatic drop in the diagnosis of late-stage breast cancers. Remarkably, there was not. Some epidemiologists have concluded that many of these early-stage breast cancers would never have progressed to cause symptoms if untreated. Nevertheless, once the diagnosis has been made, treatment is almost inevitable. In 2011, the medical journal Lancet reported on a massive Swedish study of 650,000 women, divided into two groups. Half of the women got screened annually for breast cancer. The other half was screened only at the end of the 6-year study. The researchers found 1/3 more invasive breast cancers in the group screened annually. The author's conclusion was the natural history of many screen-detected invasive breast cancers is to regress spontaneously. The moral of the story is that screening asymptomatic people for cancer is a double-edged sword. It may catch deadly cancers at a stage where they are highly treatable. But asymptomatic screening for cancer also will detect cancers that never would have caused symptoms or death. Yet these people too will be treated and potentially harmed by the unnecessary treatment.